Uh, yeah. Oh, looks like Jared just started recording. Read my mind. Thank you. Hello, everyone, and welcome to the University of Georgia's Institute of Bioinformatics seminar series. Today, I have the honor of introducing Dr. Anna Fagre, Assistant Professor of Veterinary Microbiology and Preventative Medicine at Iowa State University. Before arriving at Iowa State, Dr. Fagre was a research scientist at the Center for Vector-Borne and Infectious Diseases at Colorado State University, as well as an EIS officer with the CDC assigned to Arboviral Diseases branch in Fort Collins. Dr. Fagre was awarded a K1 Special Emphasis Research Career Award from the NIH, and her work focuses on vector-borne and zoonotic pathogens in wildlife and integrates field ecology, molecular virology, and ecoimmunology through a One Health lens. Please join me in welcoming Dr. Fagre to UGA by either using the clap reaction in Zoom or turning on your video real quick to wave a quick hello.
All right, should I go ahead and get started?
Yeah, you can go right on ahead. Thank you.
Okay, wonderful. Thank you, Guppy, and thank you everyone there at UGA, including Jara, who was very helpful in getting everything sorted. I'm sorry I can't be there with you today in person, but I'm very glad that I was able to connect with you all, and hopefully I'll be down there in person before long. I love Athens. I spent a month down there during that school doing an internship at Squidus. So yeah, Athens has a special place in my heart. But today I will be talking about some of the past work that I've done during my training and also how that folds into the work that my recently launched laboratory at Iowa State University will be digging into. So let's see. Excited to get started. I do want to start with acknowledgements because sometimes I run out of time at the end and then I rush through them. But first, want to acknowledge my PI, my advisor for my PhD, Dr. Rebecca Kading, who's still a close collaborator, and everyone who's been in and out of her lab has just been wonderfully supportive. The work I'll be talking about today has been in close collaboration with Dr. Stephanie Seifert's lab, MESAP at Washington State University, and Dr. Alexander Brown, who recently moved on but led the computational work for some of the work that I'll be talking about in the second half of the talk. Everyone in my lab, and then collaborators at Uganda Virus Research Institute and Makerere University for some of our field work in Uganda, funding support, both NSF, the VRENA group funded much of the cell culture work that Dr. Seifert's lab spearheaded, and then I'm supported by a K award, the NIH. So I want to start talking about my background in the lab and the field. And so I've kind of taken a less linear path, but this has given me some context and insights that I think has really exposed me to different disciplines in some unique and exciting ways. And while it sometimes has felt very circuitous, I like working on lots of different things, and it's helped me kind of effectively tie together concepts across systems and also across biological scales. And so I'm actually from Iowa. I moved from Iowa to Colorado, went to veterinary school at Colorado State University. And after veterinary school, I did a residency in veterinary clinical microbiology. This was largely focused on diagnostics, but also helping clinicians interpret some of the diagnostic results. Lots of molecular work, but also serology. How do we interpret these results, and what does that mean for individual animal health and herd health? The residency was coupled with a PhD program, and that's where I worked under Dr. Kading, who's featured in the picture with me up in the upper right-hand corner. And my dissertation focused on the kind of wildlife arthropod virus nexus. And so studying sylvatic transmission cycles of arboviruses with zoonotic potential and looking at those, and particularly in wildlife and even more specifically in bats, when COVID-19 happened, my work kind of pivoted and we ended up doing more coronavirus work with bats and rodents. And I missed my arboviruses. And so I was very lucky when, after my PhD, I was assigned to the arboviral diseases branch with CDC's Epidemic Intelligence Service. And so that really gave me an opportunity to apply what I had learned about arboviral circulation and diagnostics, but at more of a population level. And so thinking more about how we apply these things and learning about how public health works at the federal, state, and local levels while collaborating on research and clinical projects across sectors. And so the kind of all of these experiences together, I think, have really helped me think about how the host responds to viral infection across biological scales. And so here, if we have our little laser pointer, and if we think about biological scales as kind of this pyramid, and so at the smallest scale, we have the molecular level, and then it builds up to cellular, tissue, and then all the way up to population ecosystem level. We can see that there's increasing biological complexity as we move up in scale, but this also correlates with phenotypic changes. And so while we can measure perhaps differences in gene expression using transcriptomics, what does that look like in terms of pathology at the tissue level? And how does that impact whether or not the animal becomes viremic and can then transmit an infection onward? And how does that impact the global spread and distribution of these pathogens? And so thinking about things in this way has been really helpful for me kind of as a framework. And so I'm going to really spend more time talking about arboviruses, which is why we have our picture here of our bird being fed on by a mosquito, but to kind of pivot now into our system of choice for the day. Emerging arboviruses threaten global health security, they threaten animal health. The emergence and reemergence of many of these pathogens, chikungunya, dengue virus, yellow fever, as you can see here in this blue. So here in the Western Hemisphere, we see transmission of at least three of the four viruses that are cited here. So chick, dengue, yellow fever, and Zika. And so you can see that there's ongoing transmission and circulation of many arboviruses that have impacts on human health. And with arboviral disease, we don't necessarily have efficient, antivirals like we do with bacterial diseases. We don't have the ability to just look for an antibiotic that will treat the patient. And so we really rely on preventive care, we rely on vaccination, and we rely heavily on surveillance to really understand what's going on so that we can try to disrupt transmission. So, what is an arbovirus? Arbovirus stands for arthropod-borne virus, and these are mostly RNA viruses, with the exception of African swine fever virus, which is a weird virus. Just got done teaching my vet students about it. Breaks a lot of rules because the DNA virus that is an arbovirus for one. But mostly these are RNA viruses, and so we're going to talk about flaviviruses today. All arboviruses, by definition, they replicate in hematophagus arthropods, and so these might be mosquitoes, ticks, midges, sandflies. And they are transmitted to vertebrate hosts by a bite during blood feeding. And there are other transmission mechanisms possible. So, for example, we can see transovarial transmission in the mosquito, and this can contribute to persistence in the environment. It can contribute to overwintering. We can see direct transmission sometimes between vertebrate hosts. And so some flaviviruses are shed in the milk of an infected mammal. So there are some really odd ones. We also see aerosolization in certain circumstances. So Rift Valley fever virus, for example, sometimes is transmitted to abattoir workers just by aerosolization of those tissues. So while there are these kind of unique virus-specific context-dependent circumstances, generally when we think of arboviruses, we think of a virus being transmitted to a vertebrate by the bite of an infected hematophagus arthropod. Now let's look at West Nile surveillance here. Oh, wait, no. Oh, it's okay. One of my slides got deleted. It was supposed to say first West Nile um, amplification and transmission cycle, but that's okay. The important thing that we're going to talk about here is this is an example of how surveillance systems really target that sylvatic cycle because we do see that amplification in the environment and then we see spillover. And so one here, actually, before we do that, let's look at West Nile amplification and transmission. So generally speaking, in this particular system, we have the flavivirus, West Nile virus. It's transmitted between an amplification host in the wild. And so this salvatic cycle is ongoing, probably in kind of sometimes peri-urban areas, but also in more kind of rural areas just depending on the virus host system. But we see amplification here between the bird and the mosquito, particularly early on in the season. Sometimes we can see this transovarial transmission, this little funky little guy's mosquito larva. Um, but then what happens is generally when we see the threshold of infection get high enough in these amplification host populations, that's when we will see spillover to dead end hosts. And so with dead end hosts, they do not develop viremia sufficient to infect another mosquito, but we tend to see pretty severe disease in these dead end hosts. And so for West Nile virus, our principal dead end hosts are humans and horses. And so, historically, particularly right after West Nile entered the United States in the early aughts and began its westward expansion, dead bird surveillance actually was utilized quite heavily because some bird species, particularly corvids, experience really high mortality, and American robins and some other passerine birds seem to not experience mortality. They tend to be what we would think of as a reservoir. We use vector surveillance where these infection rate thresholds will trigger vector control measures. And then the goal here is really to disrupt transmission prior to spillover. And so I think this is just a nice kind of overview that highlights how important it is to understand the ecology of these pathogens and how understanding the nuances of these cycles really helps us identify where and when we need to, say, implement vector control measures like aerial spraying. And so thinking about this, oops, I'm going to move my little Zoom bar out of the way. There we go. So thinking about this and how we can study this host-vector-pathogen nexus kind of through a cross-scale framework, I used my big rainbow biological scale that I made in BioRender. And then here we've got, just as to kind of carry on with our West Nile example, we have our host, our mosquito, and our virus. And so this would be kind of them juxtaposed in the epidemiologic triad. And so there are factors at the host, vector, and virus level that are going to influence these interactions. And I really have been focusing for much of my work on the host and host responses, and the way and kind of the interactions between host and these other pathways, but specifically looking at compatibility and susceptibility to infection. Can the animal be infected? Can the animal transmit the infection onward? What are those innate immune responses like? What impacts them? How does stress that the animal is experiencing, for instance, impact its ability to neutralize the virus early on in infection? And so thinking about this in terms of the different kind of arms of this triad, we look at host virus interactions and we think about susceptibility, viral replication kinetics, host immune responses, and then at a larger scale, so here if we're at the population level, what's the prevalence of infection in that population? And what is the chronicity of infection? If the animal survives, does it remain persistently infected, or does it develop an immune response that is sufficient to neutralize future infections? We also, and this is, I don't do as much of this, a little bit with kind of field surveillance, but I used to work with some folks at Colorado State University that did some phenomenal work, really getting into the kind of mechanisms of infection and dissemination within the mosquito of the virus. But there are a lot of different things that we can study or think about here. For instance, overwintering, and can the mosquito remain infected while during diapause? Can it remain infected, or can it transmit the virus onto its progeny? And then here, I really love this stuff. This is thinking about how the host and the vector are interacting. And so the wildlife arthropod relationships. So we can use things like blood meal analysis, and I'll talk about that in a second here, but trying to understand the immunomodulatory impact of saliva, for instance. There are a lot of different interactions here that shape the outcomes of transmission at a larger scale. So thinking about surveillance and what we can actually learn from an engorged mosquito, I wanted to kind of talk about some of this from a bioinformatics perspective. But with a single engorged mosquito, we can actually learn a lot. So we can do pathogen testing. We can test the different parts of the mosquito to learn about whether or not the mosquito itself was infected. So we can test a leg to see if the infection was disseminated in the mosquito. We can test the blood meal, and we can see if perhaps it was just the animal that the mosquito fed on. And then if we're interested in which animal that mosquito fed on, we can extract DNA from the blood meal directly, and we can do cytochrome oxidase 1 PCR and sequencing, and use the barcode of life database, and we can actually identify the host species. So then we know who this mosquito fed on. Another really interesting and less often used, because it's rather can be a little bit tricky. But one thing we can do, and we're seeing more of this, is we can look at immune responses within the host. And so we can actually see whether or not that animal that the mosquito fed on had antibodies against that particular pathogen. And so by doing that, we can compare presence of virus, presence of antibodies, and we can say, was that an infectious contact? And if it was, then okay, that tells us something about whether that virus may be actively amplifying in the environment right now. And that helps us understand risk. It also helps us understand maybe which bird species might be worth keeping an eye on in terms of where that amplification is occurring. And so while we just talked about West Nile and birds and mosquitoes, there are many different vector-borne pathogens that have really fascinating and tricky sylvatic amplification cycles. And you might hear sylvatic, you might hear enzootic, but really what this is referring to is that amplification cycle between the arthropod and the usually wildlife host that is contributing to circulation, transmission, sometimes overwintering, migratory introductions, particularly for birds. And so it can become very complicated. But for all of these, we really want to understand what's happening here so that we can try to prevent. And really, you could insert any host here. This is very anthropocentric of me to put a stick figure of a person here, but any host that you are interested in understanding transmission towards, you could try to better understand this so that you can perhaps attempt to disrupt this spillover. And so we'll be talking here about bats, because one of the kind of main thrusts of my dissertation work that has carried onward into my current work is looking at whether bats can serve as reservoirs for arboviruses, or perhaps serve as amplification hosts for arboviruses. So we know that mosquitoes feed on bats. They share habitat, they share tree canopy, and we do have evidence of blood meal analysis, or blood meal analysis studies done in Uganda have shown that mosquitoes do feed on bats. And so there's these pieces of evidence kind of suggesting that perhaps this could be an ecological mechanism of maintenance in the environment. There's also lots of serology, lots of virus isolation, particularly some of these older studies in like the 60s, where they've actually isolated some arboviruses from bats, free-ranging bats. But also we like to think about ticks and some other ectoparasites like bat flies and bat bugs, which I'm not going to talk about as much today. So kind of focusing on this, we're really thinking here, there are so many viruses here. So if you're interested in kind of a smattering of the different bits and bobs of the literature for this, this is getting kind of old, but Rebecca and I published this earlier in my dissertation. So we're going to continue focusing here on bats and mosquitoes, and we're going to be thinking specifically about flaviviruses. So West Nile virus is a flavivirus, but we'll be thinking about bats and flaviviruses and specifically mosquito-borne flaviviruses as a model system. Um, so Zika virus's global expansion, oops, my sorry, that got a little bit covered up. But Zika virus was first isolated in Uganda in 1947 from a sentinel rhesus macaque. And so Uganda Virus Research Institute in Entebbe has this field research station where they had these towers set up and they would sample mosquitoes at different levels of the canopy. And they would also have traps or they would have caged animals at these different levels where they would then monitor these animals for seroconversion. So they had one of their sentinel macaques at one of these canopy levels develop a fever, and they um, ended up isolating Zika virus, named for the forest where this research station is located. They ended up naming the virus Zika virus. And so while initially we saw expansion and kind of circulation of Zika on the African continent, we did see expansion in the 70s and really a kind of more explosive spread in the early 2010s is really when it took off. And so we saw 2007, the App Island outbreak, and they suspect that a lot of these were probably transmitted by remake individuals, maybe traveling for like Olympic sporting events. I mean, just globalization in general is a huge driver of arboviral emergence. And so in 2015, we saw the introduction of Zika to the Western Hemisphere and the expansion in the Americas. And so since then, we've seen emergence and reemergence of Zika virus in the Americas. And so one of the questions is, where did the virus circulate during interepidemic periods, or where has it been going? And so when we see these blips, even, last year, when we see these blips and these recurrences of cases of Zika virus in humans in more urban areas, the question kind of remains, is something similar to what's happening with yellow fever virus down in South America, something similar happening here where we have this sylvatic transmission, this amplification between non-human primates and mosquitoes, and then some bridge vector species or some mosquito, perhaps either at this kind of forest edge or because of deforestation effects, or people going into the forests. That's another mechanism by which perhaps we could see spillover, but we end up seeing urban amplification cycles, and we can see onward transmission just from human to human as well, sexual transmission, transfusion events, and pregnancy. And so the neonatal impact of this virus have really stressed the public health systems in Brazil, and they've really kind of elicited a lot of attention for this virus. And so while non-human primates are generally considered the primary suspect here for our wildlife reservoir, there have been some interesting studies where some non-human primates were not like in areas where there were outbreaks, they weren't seropositive. There were kind of some interesting conflicting results. And so there are still some questions about, could there be rodents perhaps contributing to the amplification? Could there be other species in addition to non-human primates? And so one of the species here that becomes of interest is bats. And so to look here more closely at one project that we worked on during my dissertation, this was something that I think is really cool, but So subgenomic flavivirus RNA, sfRNA. So if you are looking at, here's Zika virus, here is the genome. And then when you look at this, there's this untranslated region, well, both at the 5' end and the 3' end. These untranslated regions of flaviviruses are really GC rich. And so they are full of these stem loops and these hairpin structures. And because of that, oops, sorry, got ahead of myself. So we have these really complicated stem loop and hairpin structures. And so we thought that it would be interesting to target sfRNA as a diagnostic target. And so Rebecca Kading, who was my advisor at the time, she was working with another PI, Jeff Willis, at CSU who studies sfRNA. And so they were thinking, could incomplete degradation by host exoribonucleases that usually digest the genome, the fact that they can't actually digest these really complicated knotty structures. So could this incomplete degradation lead to persistent kind of accumulation of sfRNA in the tissues of infected hosts? And so Rebecca um, brought me in as PhD student and said, let's apply this to bats. And she got an RO3 from the NIH to do just that. And so before we, I guess, kind of pivot to that, I do want to kind of take a second to introduce the idea of kind of coupling lab systems and field systems and using these coupled systems to really think about how we can better study and kind of integrate, maybe contextualize the results of some of these. So if we have the lab in the laboratory, we can generate some more mechanistic understandings of these relationships through experiments, either in vitro or in vivo. And then in the field, we tend to generate maybe more observational and correlative data. But together, I mean, we can develop when we're studying a system, whether it's bird, bat, mosquito, whatever. When you're studying a system, can you kind of continue to have this iterative data generation analysis and then integrate these data, synthesize them, refine your hypothesis, and continue to kind of build this out? And so if we apply this to the idea of bats serving as secondary reservoirs of Zika, and we want to ask, is subgenomic flavivirus RNA, sfRNA, a more sensitive diagnostic target than genomic RNA because it is resistant to degradation? We can start to, there we go. We can apply this in a field setting. And so this study that I don't even know if I did not even include the study DOI. It's embarrassing. But we published the study in Scientific Reports in 2021, and we ended up screening 439 bat samples from Uganda. And we detected Zika sfRNA actually in 3 different fruit bat species from across Uganda. And interestingly, we could not amplify genomic RNA from these same species. And so this was really interesting to us. And we were like, oh, maybe this is actually, uh, a sensitive diagnostic target that is going to actually kind of stick around longer than genomic RNA and hot dog, this is exciting. And so we were also collaborating with some other groups at CSU, namely Brian Foy's lab and Tony Schountz's lab, who were doing some Zika studies in bats. And so we partnered with them and we used some of these samples from their study to look at the persistence of sfRNA in tissues of experimentally inoculated bats to ask the question, do we see persistence of sfRNA in this more kind of experimental setting? And can we kind of validate what we saw here with our field data? Oh, there they are again. And so CSU has a breeding colony of Jamaican fruit bats overseen by Tony Schounts. And so for this particular study, Megan Miller, who led this kind of component of it, had used three different Zika strains to inoculate these fruit bats, and we ended up detecting, we could not detect genomic RNA in any of these samples. But as you can see too, like these animals were held for a month and a half after inoculation and just maintained and monitored. And then when they were euthanized, we saw, variable sfRNA in their organs. But as you can see, I mean, it really affected multiple organ systems, and we did actually detect sfRNA in the blood. And so in terms of like peripheral blood sampling, like this could, you know, potentially be a suitable diagnostic sample or target rather. We did not amplify genomic RNA from any of these. So we thought this really kind of helped to validate sfRNA as a promising molecular target. It was the first molecular evidence of Zika infection in an old world bat species as well, which we thought was really exciting. And this also raises the question, could this be applied to other flavivirus systems? And so this leads into the next set or the next kind of study that I will talk about. And this was led by Dr. Stephanie Seifert, who runs a lab at Washington State University, and her past postdoc or research scientist, I'm sorry, Dr. Alexander Brown, who has moved on. But this study was just published, golly, a couple weeks ago. And so we wanted to look at how infection kinetics or susceptibility, compatibility, how does this change based on bat species and also the strain of the virus? And so, as you may have noticed, when we are comparing these systems, these are old world bats. These are very taxonomically divergent and geographically, I mean, just on evolutionary timescales, very different than phylostomid neotropical fruit bats that are native to Central South America. So we thought it would be interesting to look at how infection kinetics and how immune responses differ. So we ended up targeting the two species that are very well characterized in terms of kind of innate immune responses. Transcriptome is pretty well characterized. And also, they are quite distantly related. So, these two species checked several boxes for us. So, Egyptian fruit bats are the reservoir hosts for Marburg virus, which is a filovirus. And so, there are pretty well characterized pathology and immune profiles for infection in this species. During Marburg infection. And there's also been a lot of historical data suggesting that they could be infected with flaviviruses, but it's kind of scattered. There's some serology, some virus isolation, and then we found sfRNA and field caught Egyptian fruit bats, but what does it all mean? So we wanted to then compare this to the Jamaican fruit bat, which is another well-characterized bat species, and the same bat species that we had used for the in vivo work on the other study. So Doctors Brown and Seifert and their team did comparative growth curves and looked at the innate immune responses in RO6E cells, which are the Rosetta cells, and AJI, which are the Artivius jamaicensis cells, and ended up seeing some pretty interesting susceptibility patterns. And so it looks like here, when we look at the Artivius cells, we don't actually see maybe like a smidge of an increase in titer, but we don't see any sort of pattern indicative or suggestive of, a strong immune response in these animals. And so this is quite interesting. But in contrast, when we look at the Egyptian fruit bat cells, the Rosetta cells, we do see pretty marked replication all the way up to 6-7 logs at 72 hours post-infection. And so when we're looking and comparing these red and blue lines, I'm sorry for not saying or pointing this out previously, but the red and blue lines indicate different strains of Zika virus. And so MR766 is this Ugandan strain that presumably would have been something that the bats were exposed to kind of from a historical evolutionary perspective. And then PRV ABC59, this blue line, this represents the 2015 strain that was isolated during the outbreaks in the Americas. And so we also wanted to compare these two strains to see if maybe there were differences in terms of the immune response. Perhaps if Rosettus had been historically exposed to this Ugandan strain, would they react differently or would they have a more kind of immunotolerant profile, phenotype, than they would when they're infected with the Asian strain, this PRV ABC59. And so this raises a lot of interesting questions about the differences here, but what's going on with the Artivius? Is there some sort of post-entry event that is allowing the virus to get into the cells, and then there's just replication is being blocked somehow within the cell? And so this raises some questions. We did look and we found that many of the purported or suspected receptors for Zika virus were present in these cells. And so that was one thing we wanted to kind of double check as well. Could it potentially be that, the receptor that's needed is not there? And there's still a lot of questions remaining about the receptors that Zika virus uses on different cell types, but the ones that have been characterized were present in these cells. And so it does really suggest that perhaps there's some sort of post-entry event occurring. The virus is being internalized and then there's no replication. And so honing in a bit more on the Rosettus cells, because we did see that really robust market replication there. Alex, Dr. Brown, and Dr. Seifer and crew did some really fantastic work. And I would encourage you, if you're interested in transcriptomics and this, to look at the paper. Alex did just an amazing job. Big kudos to him. But there's this, I really liked how he created this network analysis. And so we're able to, by looking at this differential gene expression and pinpointing, okay, which of these, when we're comparing within the Rosetta cells, we're comparing the two viral strains, where do we see the biggest differences? And where might it be that, these two viruses differ in certain ways? We've seen it in humans, we've seen it in primates. And so where in the Egyptian fruit bat, which has many elements of its immune system are unique, where are we seeing the major differences during infection with these two strains? And so we see several inflammatory cytokines that are being upregulated or downregulated. And when we look at those with the largest difference, and we see kind of which have the most connectivity, we do see these IL-6, very pro-inflammatory, CXCL8, CCL5, and CCR7. So these are all pro-inflammatory cytokines. But what's interesting is when we looked a little more closely, we did see antiviral activation or immune activation. And so I guess that slide fell out. We do see also upregulation of interferon stimulated genes. And so it appears that there are both pro-inflammatory but also antiviral pathways being activated. And so it really raises some questions about, well, at the cellular level, we can see both of these things happening. And what would it look like at the organismal level? And so if you compare this to what has been seen with these bats that are infected with Marburg, this is a figure I created in BioRender for the study. So you can find this figure in the paper also. But we were really interested in thinking about what is driving these differences. And when we can kind of start to parse out what is driving that immune tolerance in Rosetta bats that are infected with Marburg compared to humans and non-human primates infected with Marburg, can we do something similar? Can we kind of postulate or think about the potential mechanisms that could be shaping these differences. And so there have been some papers that have come out in the past couple of years looking at differential STAT2 degradation, which is a really important um, protein in the um, activation of the interferon stimulated gene response, and getting that kind of antiviral state activated in the cell in your cells. And so there are some questions here about, even species specific patterns with STAT2 degradation by Zika NS5. And so this is an area that we're really interested in exploring in the future to see if perhaps there's something going on here. But in conclusion, these in vitro systems, they offer controlled approaches, and they allow us to generate mechanistic insights that can help further spur hypotheses and shape what we're thinking about in terms of field surveillance. They can directly inform targets for field surveillance. And this study was really exciting for us because it let us really start to dig into this idea of a bat's not a bat's not a bat. There is such beautiful diversity in order Chiroptera, across order Chiroptera. And so, and a lot of people say bats are special. They have these really unique immune systems, and they do, but we see differences in their immune responses and those host virus interactions that are just as unique as each of these species are. And that can differ, as you can see with Marburg and Zika, even within a single bat species. That can differ. So the rosette bat is going to be controlling Marburg replication differently than it is controlling flavivirus replication. And so we see these pro-inflammatory pathways, and we saw, CPE, cytopathic effect. So it raises the question, are rosette bats actually unlikely to serve as a Zika reservoir? Um, and it also highlights the importance of taking this and saying, okay, what would be the next step? So the next step after doing some of, we have the cell culture work that suggests some of these kind of immune mechanisms that might be at play. We have some field data that suggests perhaps there's this, exposure happening, and using a whole organism study. So being able to study infection in the animal is really just an invaluable tool that can be used to look at tissue specific and organ specific responses. And so, for instance, in rosette bats infected with Marburg, we see hyper localized inflammatory responses and a more systemic antiviral response. And so they're able to kind of locally control the inflammation. And so, with cell culture, while it provides lots of mechanistic insights, it doesn't let us look at adaptive immune responses. And it really, it doesn't let us look at the role of T cells. And so regulatory T cells and some of these other really cool cytotoxic T cells that are involved in early clearance and early neutralization of viral infection. This remains a big question in my mind. But as you can see, I was trying to think of, okay, for my new lab group that I would state, what's kind of one question, if I could boil down our work into one question. It would be something like, how and why do some hosts get infected while others don't? Um, but also, why do some of them get sick and some of them don't show any signs at all? There are a lot of different kind of exciting questions within this, a little bit of a Russian doll nesting situation here. But we study this question across systems and across biological scales. And by doing this, we are able to take a really comparative approach and integrate these findings. And so I've been using CIPHER for my lab's kind of acronym to describe what we do. So CIPHER standing for Comparative and Integrative Pathogen Host Ecology Research. And we are establishing projects, hopefully getting started here in the next few weeks. The lab floors got polished yesterday, and so we're going to be setting up thermal cyclers today. So it's like really happening. But we're going to be launching some projects on arboviral ecology here in Iowa, throughout the Midwest, looking at host pathogen, ectoparasite networks, hopefully getting some engorged mosquitoes and learning about what's happening with some of the arboviruses here in the Midwest. And we're also really interested in how abiotic stress impacts these relationships. And so my K award focuses on how heat stress impacts some of these relationships. And I'm really interested in thinking about this, the host as this, dynamic entity that is very, how does allostasis, well, yeah, I think if you're interested in stress physiology and things that are kind of in that arena, you should reach out and we can chat. Um, I think there are a lot of really cool areas in this space. So our lab group is growing and evolving. Anna Hartwick, lab manager, Natalie Andreessen and Kit Carpenter, my first PhD students. And we're looking for teammates who like to spend time thinking about comparative health and disease, enjoy the challenge and working across systems and disciplines, and have a sense of humor and curiosity. And I say these are kind of our core research questions. I will be looking to recruit someone with bioinformatics experience probably in the next year. So if you're interested in any of this, thinking about physiologic factors, susceptibility to infection, mechanisms shaping viral persistence, and how can we use surveillance and modeling approaches to improve our predictions of viral emergence. I really love working with folks across disciplines. And so while I might not be, super heavy quantitative modeler, I work with folks that do this. And I really like working with people who are in very divergent disciplines. So please reach out if you'd like to think about how we can put some of this stuff together. Yeah, I think that's it. Thank you so much. And I'll take any questions. I guess here, this is my Iowa State email, but I'll also, I know I let Guppy know that I would love to chat with anyone who wants to talk more, either about ideas or especially students, especially trainees. So please reach out if you would like to, and we can chat. Thank you.
Well, I'll go ahead and start us off with questions if that's all right. So the first question I have is, do all flaviviruses have this like subgenomic flavivirus RNA region? And if so, is it sufficiently unique to differentiate between pathogens, or is it too conserved because it's like that very GC rich region?
Oh my gosh. These are, that's just I just got goosebumps. What a good question. What an amazing question. So interestingly, we do see some level of conservation, but you would honestly, I mean, I expected there to be a much greater degree of conservation because of how GC rich they are. What's really interesting is you can see that the actual structures themselves, so the kind of number of stem loops and hairpins, there are very unique constellations of those hairpins structurally based on which arthropod vector transmits them. But we see sequence differences. So it's like the structural conservation is there within like the mosquito clade or within the tick-borne clade, but it's not as easy. So I didn't include these data, but I've been working with another group who's been trying to. I designed an assay that should, in theory, amplify West Nile and St. Louis encephalitis virus, sfRNA, and it was not as easy as I thought it would be. My first idea during PhD, we were like, well, can we design some sort of like pan sfRNA assay? I mean, the best way to do that, you really kind of have to start upstream of the kind of stop codon. And so if you want truly just 5' or 3' UTR sfRNA, I think it would be possible, but it would take some kind of creativity and I don't know, but that's definitely on my list. And I want to try to optimize this bird flavivirus assay a little bit better. So yeah, I think you're uh, you're definitely thinking the right like uh, our brains are similar. This is yeah.
Very cool. Thank you. If no one has questions, I have another question. Um, so mosquitoes, correct me if I'm wrong, but I thought that mosquitoes were fairly picky about their hosts. Are there bat-specific mosquitoes, and like how do they contribute to like these arbovirus like transmission chains?
That is also, oh my gosh. Guppy, your question, these are just like exactly the things that I spend all my time thinking about. So I think there are a lot of questions remaining in my head actually about bats and mosquitoes and how this transmission happens. I was actually like sketching out some ideas last night for like measuring infection by ingestion because this is also another way that we think about potentially, could an animal seroconvert and not become actively infected, but just, be exposed during feeding. And so we know that bats eat mosquitoes. And so this is one purported mechanism of transmission. But the actual, in terms of like host preference, mosquitoes, there are some mosquitoes that are, strictly ornithophilic and they really don't want to mess with mammals at all. So like Culicida that really amplifies Eastern equine encephalitis virus out there on the East Coast, very ornithophilic species. And then the way that triple E gets into mammals is via these bridge vectors, which are usually 80s or Culex, but usually 80s. And so I think that when we think about in terms of bats and mosquitoes, I think that that's one big missing piece of the puzzle for me is trying to understand like feeding preferences and um, within like a local ecological context too. So if we're looking at Zika in Ugandan bats, trying to tie in some of the blood meal analysis work that's been done there in Uganda and say, okay, does this match up? Now it can be very difficult to trap and gorge mosquitoes, even in a great, perfect, pristine setting like a golf course or something, where there aren't challenges like caves and swamps and rivers and things that you have to try to navigate. But yeah, I think the host preference and the vector ecology stuff, I mean, it can change over the course of a season. Rebecca did some work during her postdoc showing Culex mosquitoes and how they eventually shift towards more mammalian feeding over the course of the season. And so that probably is one of the things driving spillover to humans and horses. So yeah, that's a great question.
Well, do we have any other questions from the group? I don't see any in the chat or raised hands. So um, if everybody could go ahead and give us another round of applause and thanks to Dr. Fager. Thank you for coming to speak with us.
Thank you so much, everyone. Have a great weekend. And please reach out if you'd like to chat. Thank you.
Great. Thank you.